When a sperm cell begins to fertilize an egg, an embryo begins to form. Attached to an embryo is a yolk sac. Primordial germ cells are found in the walls of the yolk sac, but eventually will move to the dorsal boundary or lower back of the embryo and multiply to form the egg and sperm cells. Eventually, we will only be able to find germ cells in the ovaries and testes. Germ cells are the only cells that can divide in a way that causes the chromosomes to split in half. Chromosomes are composed of DNA and proteins and contain the primary genetic information of the cell. Chromosomes are what makes up our traits. We receive half our chromosomes from our mother and half from our father. So, in total, we have 23 pairs of chromosomes. A normal cell containing all 46 contains the diploid amount. A cell with half, just 23 chromosomes, is called the haploid amount. These cells are the gametes produced by germ cells. When a normal cell divides, it is called mitosis. The first stage of mitosis is interphase, and it is the time between cell division. DNA is present in thin strands called chromatin, and during the S phase of interphase, DNA replicates. The next stage is called prophase, where the thin strands of DNA, the chromatin, form chromosomes. The nucleolus disappears, and the centrioles begin moving to opposite ends of the cell. Microtubules extend from the centriole and attach to the centromeres. The third phase is metaphase. The chromosomes align in the center of the cell. Microtubules help pull apart the chromosomes. Fourth is anaphase, and that is where the chromatids separate and then they are individual chromatid, which they call chromosomes. And the last phase is telophase, and that is where the chromosomes begin to unravel and a nuclear envelope forms. The nucleoli form, and then you end up with two cells. Meiosis is the division of germ cells. Meiosis produces 4 genetically different cells, while in mitosis we learned that it produces 2 identical cells. During interphase 1, the chromatin replicates, so you have the same chromatin twice to form a chromosome. During prophase, the chromosomes from your dad and mom are coming together and wrapping around each other really tightly, swapping segments with each other called crossing over. During metaphase, they align in the middle of the cell and spindle fibers attach to each centromere of the chromosomes. During anaphase, homologous chromosomes move apart to opposite sides of the cell. Then during telophase, new nuclei will form and the cell will divide. After we go through meiosis 1, then we will begin to start meiosis 2. Meiosis 2 starts with prophase 2. That is where each chromosome consists of 2 chromatids. After prophase 2, we have metaphase 2, where the chromosomes align along the center of the cell. Then we have anaphase 2, where the chromatids separate, and each is now called the chromosome. During telophase 2, new nuclei form around the chromosomes, and then the cells divide to form 4 new cells with a haploid number of chromosomes, which means they have 23 chromosomes in it.